Meiosis is a type of cell division that results in 4 daughter cells and is split into 2 consecutive cell divisions, meiosis 1 and 2. In interphase, the pair of homologous chromosomes duplicate. Chromatin is uncondensed, and there are 2 centrosomes. Meiosis 1 separates homologous chromosomes. The process starts with prophase 1, which is the longest stage of meiosis. Chromosomes and the nucleolus begin to condense, and homologous chromosomes loosely pair up into tetrads. This pairing is known as synopsis. Crossing over occurs, which is where the DNA molecules of non-sister chromatids are broken and rejoined. Chiasmata are the X-shaped regions where crossovers have occurred. These mixed chromosomes are called recombinant chromosomes. The centrosomes begin moving to the opposite poles of the cell. Microtubules then begin forming the mitotic spindle along with the centrosomes. The nuclear envelope begins to dissolve. Microtubules then attach to the kinetocores at the centromere of each homologue, and these homologous pairs will move towards the metaphase plate. In metaphase 1, pairs of homologous chromosomes are arranged at the metaphase plate, with 1 chromosome in each pair facing each pole. Both chromatids of one homologue are attached to kinetochore microtubules from one pole, while both chromatids of the other homologue are attached to microtubules from the opposite pole. Gregor-Mendel's law of independent assortment is applied here, which states that each pair of alleles separate independently of each other pair of alleles. In other words, the homologous chromosomes line up randomly at the metaphase plate, allowing for the independent assortment of alleles. Anaphase 1 separates homologues by breaking down the proteins responsible for sister chromatid cohesion. The spindle apparatus guides the homologues towards opposite poles. Sister chromatid cohesion persists at the centromere, so that chromatids still move as a unit towards the same pole. During anaphase 1, Mendel's law of segregation is applied. The law of segregation states that 2 alleles of a trait separate or segregate during gamete formation. In anaphase 1, the pair of homologous chromosomes separate, essentially separating the alleles into different cells. Telophase 1 and cytokinesis occur simultaneously. Each half of the cell has a haploid set of duplicated chromosomes. Each chromosome has 2 sister chromatids that include regions of non-sister chromatid DNA. A cleavage furrow forms in most animal cells, and a cell plate forms in plant cells. No chromosome replication occurs between meiosis 1 and 2. In some species, nuclear envelopes reform around the chromosomes in the 2 daughter cells. Next, meiosis 2 begins. In meiosis 2, sister chromatids are separated. The first part of meiosis 2 is prophase 2, in which a spindle apparatus forms. Towards the end of prophase 2, chromosomes start to move to the metaphase 2 plate. If nuclear envelopes reformed in telophase 1, they now fragment in preparation for meiosis 2. In metaphase 2, the chromosomes are positioned at the metaphase plate. The 2 sister chromatids are not genetically identical because of crossing over in meiosis 1. The kinetocores of sister chromatids are attached to the microtubules extending from opposite poles. This leads to anaphase 2, where sister chromatids separate because of the breakdown of proteins holding their sister chromatids together at the centromere. These chromatids move towards opposite poles as individual chromosomes. The last part of meiosis is telophase 2 and cytokinesis. At this stage, chromosomes begin to decondense and nuclei reform in the daughter cells. This phase results in the formation of 4 haploid daughter cells that are genetically distinct. In males, 4 sperm cells produce, whereas in females, only 1 egg cell is produced, and because it absorbs the other 2 daughter cells in a process known as oogenesis. Sperm cells develop tails for locomotion, while egg cells do not. By the end of meiosis, 1 diploid parent cell splits into 4 haploid daughter cells.